Next, we'll be looking at specification number 8. This involves investigating how light intensity, carbon dioxide concentration, and temperature affect the rate of photosynthesis. These are the same factors we have just studied, and now, through these experiments, using submerged aquatic plants and hydrogen carbonate indicator solution, we'll find out how well our theoretical understanding matches real observations. The diagram here shows the setup we'll use, including a lamp, a test tube with the submerged plant, and a thermometer. So let's begin. So, we already know that during photosynthesis, plants release oxygen gas into their surroundings, right? Now, here's something interesting: the amount of oxygen produced can actually tell us how fast photosynthesis is happening. Now think about aquatic plants, like Elodie or Kabomba. When they photosynthesize underwater, the oxygen comes out as tiny bubbles. If we watch closely, we'll see these bubbles rising to the surface. And guess what? By simply counting how many bubbles appear in a minute, we can estimate how fast the plant is photosynthesizing. More bubbles. That means the process is happening faster. But what if we want a more accurate result? Well, there's another way. Instead of counting bubbles, we can place an inverted test tube over the plant and collect the oxygen gas directly. This way, we measure the actual volume of gas, which is more reliable because bubble sizes can vary. Using this setup, we can test how different factors, like light intensity, temperature, or the amount of carbon dioxide, affect the rate of photosynthesis. It's a great way to connect what we've learned in theory with real, observable results. In this experiment, we'll be using something called hydrogen carbonate indicator. But before we get into that, let's first understand what it is. Hydrogen carbonate indicator helps us see how much carbon dioxide is present in a solution. Carbon dioxide, when it dissolves in water, becomes slightly acidic, and that's what allows this indicator to work. Now, we can use this indicator to investigate photosynthesis in aquatic plants. Since carbon dioxide is one of the main ingredients for photosynthesis, measuring how quickly it's removed from the water gives us a good idea of how fast photosynthesis is happening. So how does it work in practice? We place an aquatic plant in water that contains the hydrogen carbonate indicator, and then we watch for any color changes under different conditions. For example, under bright light, photosynthesis happens faster, using up more carbon dioxide. As a result, the indicator changes from red to purple. But if the plant is in the dark, it can't photosynthesize, though it still respires. Respiration produces carbon dioxide, so in this case, carbon dioxide levels rise, and the indicator turns yellow. In short, by observing how the color changes, we can understand how different factors like light, temperature, or carbon dioxide levels affect the rate of photosynthesis. Now let's look at this chart that shows how the hydrogen carbonate indicator reacts to different levels of carbon dioxide and what that tells us about what's happening inside the plant. Starting at the top of the chart, when carbon dioxide levels are the highest, the indicator turns yellow. This usually means the plant is in the dark or not getting enough light, so it's respiring more than it's photosynthesizing. As a result, more carbon dioxide is being produced than used, and the solution becomes more acidic. Next, when the carbon dioxide level is still high but not at its peak, the indicator shows an orange color. In this case, respiration is still greater than photosynthesis, but not by as much. At the middle point, when the carbon dioxide concentration is about the same as it is in the atmosphere, the indicator appears red. This tells us that photosynthesis and respiration are happening at roughly equal rates. The carbon dioxide being used by photosynthesis is balanced by the amount being released through respiration. Now, if carbon dioxide levels drop even lower, the indicator turns magenta. That means the plant is photosynthesizing more than it is respiring, so it's using up more carbon dioxide than it's producing. This leads to a more alkaline solution. Finally, when the carbon dioxide level is at its lowest, the indicator becomes purple. This indicates that the plant is photosynthesizing much more than it is respiring. Photosynthesis is happening at a very high rate, often under strong light. So overall, this chart helps us understand how the color of the hydrogen carbonate indicator reflects the balance between photosynthesis and respiration, which we can use to measure how environmental conditions like light intensity or temperature are affecting the rate of photosynthesis.